Hello AP Biology students, we're going to go over the 5th section in Unit 4. And in this section, we're going to review the steps of mitosis. Remember, up until this point, we've been talking about the whole cell cycle, and before M phase comes interphase. During interphase, we're going to have G1, S, and G2 phases, and this is where the cell is going to go through normal functions, grow, and get ready for mitosis. During S phase, we're going to see the DNA be replicated or duplicate itself. This is in order for when mitosis occurs that each new cell is going to have the same exact genetic code. Before I actually go into the steps of mitosis, we need to make sure we understand that M phase is two different parts. It's mitosis and cytokinesis. One of the biggest misconceptions during this whole unit is that M phase is just mitosis. Mitosis is specifically the division of the nucleus, while cytokinesis is the division of the cytoplasm. Both mitosis and cytokinesis are within the M phase, but they are two separate things. The first step in mitosis is called prophase, and this is where the chromosomes are going to condense. So again, up until this point, chromosomes have not condensed yet. That's another big misconception, is that during interphase, the chromosomes are uncondensed. You can see the chromosomes here condensing into that X pattern that we typically identify them in. But we see something else forming. These are going to be called mitotic spindles or spindle fibers. These spindle fibers are going to be specifically the proteins or just things that move the chromosomes around. At this point in mitosis, the nucleus or the nuclear membrane starts to break down. The nucleus needs to break down so the spindle fibers can attach to the chromosomes and then start to orient them in the cell. Just as a review, the main things that happen here are the chromosomes condensing and the cell is starting to get ready to divide the nucleus. The next step of mitosis is called prometaphase. You actually might see some textbooks label this as late prophase, and some textbooks don't even include it as a separate step. I like to go over prometaphase in my AP biology classes, but in my regular biology classes, I just kind of wrap this into the metaphase step. During prometaphase, the kinetochores are going to appear. These are just protein structures around the centromeres where the spindle fibers are going to attach. It's important to remember that at this point, there is still no orientation of the chromosomes, which means they're still kind of just randomly found within the cell. During the next step, metaphase, the chromosomes are going to align in the middle of the cell. When I think metaphase, I think middle. You can see here the chromosomes aligning in the middle, and they have that orientation because the spindle fibers have moved them to that location. After metaphase, we're going to see anaphase, and this is where these sister chromatids are going to be split apart. This is due to the spindle fibers shortening and then literally just kind of ripping these chromosomes in two. You can see the chromosomes or the chromatids of each chromosome being split apart and then traveling to the poles or the opposite ends of the cell. Remember, we're trying to get the correct amount of chromosomes in each cell. We began with 4 chromosomes, and we want each cell to have 4 chromosomes when mitosis is done. That's why during metaphase, we need to have the chromosomes align correctly, because if that doesn't happen, each cell might not get the correct amount of DNA. During telophase, the spindle fibers are going to start to disassemble, the nuclear envelope or nucleus starts to reform around the chromosomes, and the chromosomes start to unwind. This is where we start to kind of see 2 new cells be formed, but again, we still don't have 2 new cells. We're just 1 cell right now. This is where we technically see the nucleuses, again, the nucleus here and here start to reform, and we're starting to see that whole process come to an end. This is technically where we see mitosis end, since we're starting to see the 2 nucleuses reform. It's not until cytokinesis do we see M phase ending. M phase, again, is mitosis and cytokinesis. Cytokinesis is just the division of the cytoplasm. So once cytokinesis is done, then M phase is done. But cytokinesis is different than mitosis. It's still in M phase, but it comes after mitosis, and it's just the division of the cytoplasm. At this point, each of the cells should have a nucleus full of the correct or starting amount of chromosomes. I like to mention here, when mitosis occurs without cytokinesis, you're going to see a cell with 2 nuclei. Since again, cytokinesis is the division of the cytoplasm, we don't have that here, so we have mitosis creating 2 nuclei, but not 2 new cells. Whenever cytokinesis occurs without mitosis, you're going to see a cell without a nucleus, since we didn't double the nucleus. We'll still have that original cell, but we also have a cell here without a nucleus. Now, the end result of mitosis is going to be 2 genetically identical diploid cells. And this should, again, make sense because we're creating body cells. We don't want to have genetically different cells within your body cells. Typically, about 95% of your cells are in interphase right now, and only about 5% are going through mitosis. The reason why only 5% are going through mitosis is we don't want to continuously grow every single cell or every single organ or tissue. That could lead to cancer, which is uncontrolled cell growth. So we try to regulate within our bodies how much our cells go through mitosis. So in total, only about 5% of your cells right now are going through mitosis, since we don't want a lot of your cells to be dividing all at once. We're going to end on a subpar meme. Nobody cells performing mitosis. Let's split up, gang. It's the best I could do right now.